Today we are going to start a new interesting chapter, that is biomolecules. And in the previous session, as you know that we have already completed the DRS, right?
Yes, sir.
So have you completed the homework assigned for the same?
No, sir.
No, why?
I forgot, sir. Today I complete, sir.
Okay, so can you tell me that how many homework is pending from your end? No
What,
idea.
sir?
How many homework pending from your end, as we generally assign the homework to you, right? So how many homework is pending from your end? Any idea?
Many are there, sir.
Many are there. Why not completing?
I have to complete, sir.
When? When you will complete? Any idea? No. So see, just my simple request to you, whenever the session is there, and you know that in that sessions there is a class assessment and homework, right? So
Yes,
class
sir.
assessment generally we used to do during the live session, and then remaining homework is assigned to you as a homework. So you can try to finish, because homework hardly, it's just 10 question, matter of 10 question, right? And it will take hardly 10 to 20, 10 minutes might be, isn't it? So what my request is, whenever we assign homework and assessment, class assessment is done from our end, because we generally help them to complete the class assessment. But homework is from your end. So try to complete your homework on the same day, so that will be not going to create a lot of burden on your, that, oh my God, so much homework is pending from my end, right? And this create a bad impact when suppose we generate a result of your monthly report, progress report, so that time it create bad impact on your result. So never do that, okay? Whatever the homework or class assessment, you have to be to complete on time, so that will create a good impact of your result of every month. As you know that there is a PTM will also be conducted, right? So whenever the PTM will conduct, in that time your results should be good in, otherwise how you will come in the PTM, like, oh, my result is not so bad, so why should I come in the PTM, right? So don't create such kind of, you have to be complete your homework on time. That is what my suggestion to you. Clear?
Sir.
Okay, so I think so. Anushri is not going to join. So now just you and me in this session. So whatever the doubt if you have during the session, you can ask immediately. Don't keep it like, oh, okay, it's not required. No, whenever you have doubt, you have to ask immediately. Clear?
Yes, sir.
Okay. So now let's start with this chapter, that is, as the chapter name is biomolecules. Why we need to study these biomolecules? Do you have any idea? Or do you have any idea that what we are going to learn in this chapter, biomolecule?
No, sir.
Oh my God, someone singing from your side?
No, sir. It is TV sound, sir.
Okay, okay, all right. I thought someone singing.
No, sir. No, sir.
Okay, all right. So yes, tell me what you understood from this word biomolecules.
Sir, it is biochemistry, sir.
Okay, biochemistry. Why your word biochemistry? Why not biomolecules?
No, sir. Molecules are mostly comes under chemistry subject.
Oh, so this is the logic that molecule means it's from biochemistry.
Yes, sir.
Okay, but let me tell you, biomolecules means basically those molecules which is biological in origin, right? That means of though there is a chemical, but it is not involved the pure chemistry. Like it's similar to organic chemistry, all the organic related. So biomolecules, especially we are going to deal with the carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. Okay, what? So in this chapter we will deal with C, H, O T N S. Okay, so these are the important molecule. C for carbon, hydrogen, oxygen, phosphorus, nitrogen, sulfur. These are the important chemical component which is the part of biomolecules. So basically it is a part of biochemistry, but biomolecule means its origin is biologically. Clear?
Yes, sir.
Now here see the combination of these 6 words and with the beautiful questions that I am a building block of all the living organisms. Who am I? Yes, tell me. Who am I?
The molecules, sir.
Now, DNA is the building block of all the living organisms. DNA,
Yes, sir.
right?
Yes,
And
sir.
that DNA contain nucleic acid, carbohydrate, that means of in the form sugar. Then there is a lipids and protein. So you know that DNA, simply how actually protein is created, because DNA when translated it form mRNA, sorry, transcription, not translate. When DNA transcript, it form mRNA, and when mRNA translate, okay, it form proteins. Clear? That means of DNA is the building block of all the living organisms, which includes nucleic acid. So nucleic acid means what? That is pentose sugar over here. This is a pentose sugar. This one is the pentose sugar. This is phosphate group, and this is the molecule. So what happened? They form nitrogen bases. So it form a complete nucleic acid. This is the carbohydrate. So carbohydrate means what? In the form of sugar. So monosaccharide, disaccharide, trisaccharide, tetrasaccharide, like that. Lipids means glycerol, and proteins. Lipids means not only the glycerol but all the fatty substances, and the proteins. These proteins, how the proteins are made up of? So proteins are nothing but it's a chain of amino acid. So here you can see this is the one amino acid, this is another amino acid, this is another amino acid, and when they form a long chain, it is called polyproteins. Clear? So it's a long chain of proteins. Clear? So who is the building block of all the living organisms? DNA.
DNA.
Clear? Now, as you know that the living systems are made up of various complex molecules. That complex molecules are what? Nucleic acid, carbohydrate, proteins, and lipids. Right? Now it is very much clear from the previous slide, isn't it?
Yes, sir.
Yes. So when I say organic compound, there are basically the cell component, all the cell component, living cell component, like your body cells, my body cells, plant body cells. All these cell components are made up of collection of molecule called the cellular pool. These are the collection of molecules, and which consist of both inorganic, organic, include some salt, minerals, and water. That means of all the living organisms are made up of from organic, inorganic, salt, and minerals and water molecules. But the organic compound include the carbohydrate. Organic compounds include the carbohydrate, lipids, amino acid, proteins, nucleotide, some kind of hormones, and even little bit amount of vitamin also. So some organic molecules remain in a colloidal form in the aqueous intracellular fluids, and other exist in the non-aqueous phase like lipid membrane, cell wall, right? So that means of all the, so here you can see the cell membrane. So this is cell membrane, the component that you have already studied, that the cell, how exactly made up of. So the cell wall is basically, they have cell membrane, like here you can see how the structure of it. This is an integral protein, this is channel proteins, then alpha helix proteins, peripheral proteins, because it present towards the periphery, so it's a peripheral protein. Cholesterol over here, carbohydrate, globular protein. So these are the cell membrane, right, which we might be also study about the fluid mosaic models, right?
Yes, sir.
So that we have already studied in the previous chapter. Now here you can see that all the compound or the molecule present in the living cells are called the biomolecules. All the compounds or molecule present in the living cell. So living cell, what kind of in the living cell, what are the important things are present? So you can see in the living cells there is nucleus, then there is a mitochondria, chloroplast, right? These are the different type of cell organelles which is present inside the living organisms, right? So it includes large macromolecules. And what are those macromolecules? You can see here that include the proteins, then polysaccharides, lipids, and nucleic acid. Now these biomacromolecules are polymers. Biomacromolecules are polymers. Why it is polymers? Because it was made up of from a group of chain. It is not just made up of from one particular amino acid. It is a group of chain. That's why it is a polymer. And these are large sized, high molecular weight, complex molecule that are formed by the condensation of biomolecules. So when these are condensed, they form a biomolecules. So you can see the typical structure. This is the plant cell wall, and inside the nucleus there is a chromosome, a particular segment of chromosome. There is a DNA. So DNA is what? It is a particular segment of chromosome. Clear? Then inside that DNA, it is made up of from nucleotide. Nucleotide means there is a phosphate group, then followed by there is phosphate group, then followed by there is some kind of sugar, and then followed by there is uh, hello, I think so one more student joined. Hello. Yes, Pradyum, can you hear me? Pradyum, Pradyum. Oh, just a minute. Sorry, Arti. Actually, Anusika informed that she has exam and that's why she is not going to join today. Okay, all right. So, what I said about that in a particular cell means, you know, that our body cells are made up of crore and crores of cell, because you have already studied about that all the living organisms are made up of from cell. So what happened, that cell, a particular region, that is nucleus inside the cell, contain the chromosomes, and that chromosomes, when you enlarge that view, chromosome is a long thread-like structures, right, highly coiled. So a particular segment of the chromosome, that is the DNA, right? Like suppose a long thread is this much. So when this much portion considers a one segment of DNA. And in that segment of DNA, it contain phosphate group, then followed by pentose sugar, phosphate group, pentose sugar, and nitrogen bases. So it is called nucleotide. Clear? Now similarly, there is a plasma membrane. So inside that cell there is a plasma membrane, and that plasma membrane basically having a proteins, and that proteins is made up of from amino acid. So, similarly, cell wall is there, cellulase, and this cellulase is made up of from sugar. That means of all the component of cell, basically we can call it is biomacromolecules, isn't it? Yes. So they are condensed. Now, how to analyze the chemical compositions? Like suppose I wanted to know that what is there in the liver. Okay, so I want to analyze the chemical component of liver or carrot. What I will do? How to analyze the chemical constituent? How I can find out that what kind of chemicals are present in that liver? What kind of chemicals are present in the plant cell? So for there, what I can do? I will take the liver or tissue in the motile and pestle. Here you can see this is the motile and pestle, isn't it? So this is a very common motile and pestle. Generally in our village, we generally saw this kind of, isn't it? So this is a common motile and pestle. You have to just crush the any living tissue in this motile and pestle. After crushing, make it slurry by adding the trichloroacetic acid using a mortar and pestle and obtain a thick slurry and strain through a muslin cloth, or there is a cheesecloth is also available. So you can use a cheesecloth or muslin cloth or any simple cotton plant. Just simply you can make a to get the fractions, that means of the trichloroacetic acid, and the cell mass gets separated. Like it's similar to when we use a mace for tea separations. no? So that you with the help of that cotton you just have to make separate. And after making the two fractions, one is called the filtrate. Like suppose this is a methylene cloth. So inside the lower here you can see this is called the filtrate, or more technically the acid soluble pool, and the second is called the retenate. This is the retenate which is remain or the acid insoluble fractions. So that insoluble fraction is not required for us, but the thousands of organic compounds in the acid soluble pool is there. That means of inside this is your supernatant. So inside this supernatant there are thousands of organic compound present in this uh, soluble pools. Okay, now the next stage is to analyze that what kind of chemical component present. So what you will do, simply when you analyze under the any either you can use the spectrophotometer, or you can even you can use the HPLC, GC, GCMS mass spectroscopy. These are the different type of instrument. Through that you can able to analyze that what kind of chemical constituents are present in that, right? Now here you can see on the basis of that all the chemical compounds are classified into two category: primary metabolites and the secondary metabolites. Now what is the primary metabolites and what are the secondary metabolites? So when I say primary metabolites, it's similar to what that most plant, fungi and other microbes synthesize a number of organic compound, and that compound are called the metabolites, right? That compounds are called the metabolites, which are intermediate or the product of metabolisms. You might have seen when you visited, have you visited any particular forest or we can say jungle? Have you ever visited? No, yes,
Yes, sir.
yes. Have you visited any particular forest?
Yes, sir. In my village, I visited, sir.
Okay. So have you ever see and what is the season that have you visited in the winter season or rainy season?
Rainy season, sir.
Okay, so on the rainy season, you might have seen that there are small fungi growing on that ground. Either it can be a mushroom type of fungi
Yes,
growing,
sir.
right?
Yes, sir.
So
Yes, sir.
that mushroom might be, they releasing some kind of chemical constituents, like, kind of some poisonous substance they're releasing from their body, right? Now these poisonous substances are the secondary metabolite. These are the primary and secondary metabolites. Okay, these primary and secondary metabolites help plant to protect from the other grazing animals. Like suppose any mushroom produced secondary metabolite which is poisonous for any other animal. So animal try to avoid that poisonous mushroom, isn't it? So it is a kind of protection, but it is nothing but it is a metabolites which can be helpful for human beings, right? So therefore primary metabolites are involved in the growth, development and reproduction of the organisms. See, what are the primary metabolite and what are the secondary metabolites? It's very simple. Like inside your body there is a lot of hormones, then we can say the neurons or the other molecules which are generally synthesized in your body, but that molecules is required for your growth, isn't it? Yes and no, Arthi?
Yes, sir.
Yes. So primary metabolite are those we can simply say metabolite that are involved in the growth, development and reproduction of the organisms. Means it is always required for our body. But secondary metabolite are those metabolite which is not required for the growth, development of an individual, but it is just required for the protection. To get protect from the other enemies, there is a secondary metabolites, right? Or it is not required for secondary metabolite are those metabolite which is not required for the growth and development. So here you can see ethanol, lactic acid and certain amino acid are come under the primary metabolite which is required for the plant. Now secondary metabolite are typically organic compound produced through the modification of primary metabolite, and these metabolites are not required for plant, but that can be required in the sense of for protections. Clear? These are the primary and secondary metabolites. Apart from that, other here you can see that some micro and macromolecules is also required for the body, the growth for any individuals. So micromolecules element on the basis of presence and requirement in plant and animals, there are the grouped into a major and minor component, major and minor elements. Now what are the major elements? Calcium, then potassium, phosphorus, then magnesium, sodium, magnesium, sulfur, potassium, nitrogen. These are the major 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 micromolecules. There is a minor micromolecules which is iron, copper, cobalt, manganese, molybdenum, zinc, iodine. These are the minor component. Means it is required in a very small quantity, but the effect is very high, right? And these are the percentage which is written over here. Now other, on the basis of the functions, they may be of the following types. Like we can see the framework element which includes the carbon, oxygen and nitrogen. Protoplasmic element which includes the proteins, nucleic acid, lipids, chlorophyll and enzymes. Balancing element like calcium, magnesium and potassium, right? So these are the micromolecules. Now, and their importance, like oxygen, carbon, then hydrogen, nitrogen, calcium, silicon, along with their earth crust, and in the human body their requirement, oxygen requirement is 65%, carbon requirement in human body is 18%, then there is hydrogen 10.5, nitrogen 3.3, calcium 1.7 and silicon is negligible. So all these elements are must for any body growth, right? Now biological compound, or we can say organic and inorganic compound. So water, proteins, nucleic acid, carbohydrate and lipids, these are the 5 important uh, components in any cell. So maximum compound in the cellular pool is water, while the maximum organic compound is proteins. So you can see maximum This is the organic compound which is maximum about 15%, and minimum is lipids. The ratio of organic and inorganic compound to the cell is 9.1, 1.9. That means of 1% is your inorganics, so 9% is your organics. So these are the ratio of biological compound. Okay, now we just studied about that what are macromolecules, primary and secondary metabolite. We also studied about the organic and inorganic component. Now, as I told you that the biomolecules are the complex substance, and it includes nucleic acid, it includes nucleic acid, then second it includes the carbohydrate, carbohydrates, third it includes the proteins, and fourth is lipid, isn't it? So we will first focus on carbohydrate in details, that what is carbohydrate and all. So carbohydrate are naturally occurring compound having the very general formula is CXH2OX. Sorry, OY. Okay, C2H CXH2OX. This is the general formula of the carbohydrate. Okay, and these carbohydrate which have a constantly produced in nature and participate in many important biochemical reactions. Like very simple one, I can say glucose. What is the formula of glucose? C6H12O6, right? These are the simple formula of glucose, right? Similarly, there is a ribose. So glucose, ribose, then there is a cellulose and starch. Cellulose and starch. Clear? These are the four important carbohydrate process components. Clear? Now, it is formed during the photosynthesis. When this carbohydrate is formed, you know that the simple formula of photosynthesis. How photosynthesis? So, you know, very simple chemical reactions you have already studied in from the 7, 8, 9, 10th, right? A very simple formula of photosynthesis is plant absorb carbon dioxide from the atmosphere, isn't it? It also absorbs water from the ground with the help of chlorophyll pigment like sunlight, okay, and it form C6H12O6, which is glucose plus water and plus O2 is released, isn't it? Okay, so these are the simple equations. So what is the product? Product is glucose. We are interested in glucose, so product is glucose. So here you can see that this carbohydrate is formed during the photosynthesis, and it exists only in 1% but constitute 80% of the dry weight of the plant. That means of this photosynthesis exists only in 1%, but their major constituent is 80% of the dry weight of the plant, right? So these compositions having carbon, hydrogen, oxygen, and what is the ratio? This is the ratio which is represented here. So please remember this glucose and the ribose formula. All right. Okay. Now, classification of carbohydrate. So as you know that the first carbohydrate generally prepared with the help of photosynthesis. Now this carbohydrate are classified based on the presence of their sugar molecules. So here you can see carbohydrate monosaccharides, oligosaccharides and polysaccharides. So basically it is classified into three types, and that includes the monosaccharide, oligosaccharide and polysaccharides. Now here disaccharide is what? Sucrose and maltose. Trisaccharide is what? Raffinose. Right? These are the important compound, and you have to also remember that sucrose and maltose is disaccharide. Raffinose is a trisaccharide. Clear? Okay. Now structural or polysaccharides like cellulose, storage polysaccharide, starch and insulin. So structural polysaccharides are cellulose. Where it is present? It is present in the plant cell wall, because plant cell wall is made up of from cellulose, isn't it? So that's why this structural polysaccharide is a cellulose which is present in the plant cell, and this glucose or the structure of carbohydrate which prepared by the plant, they can also stored in the form of polysaccharides. So these are the starch and the insulin. Okay, so insulin starch is the storage polysaccharide which is generally stored by the plant in their body. So monosaccharide, trisaccharide, there is a triose. What is triose? That is called the glyceraldehyde. Tetrose, that is erythrose. Pentose, that is ribose and xylulose. Hexose, which is include the glucose, and fructose. Hexose means glucose and fructose. And there is a heptose which is called the pseudoheptulose. Pseudoheptulose. Yes. Okay. All right. Now, as I told you that carbohydrate is classified into three categories: monosaccharide, oligosaccharide and polysaccharide. Monosaccharide is just a simple single molecules, so monosaccharides, which includes further, that is a triose, tetrose, pentose, hexose and heptose. Right? Now trioses, which include the glyceraldehyde, tetrose, which includes the erythrose, pentose, example is ribose and xylulose, hexose include glucose and fructose, heptose includes the pseudoheptulose. That you have to remember. Oligosaccharides is classified into two types: disaccharide and trisaccharides. So sucrose, maltose and raffinose. Polysaccharide is structural polysaccharides, cellulase, and the storage polysaccharide, which include the starch and insulin. So in short, what you have to remember? You have to remember the monosaccharide, oligosaccharide and polysaccharide. Monosaccharide is classified into five category: triose, then dextrose, then there is tetrose, then there is a pentose, hexose and heptose, along with their example. Are you getting my point? Okay. Oligosaccharide is two types: disaccharide and trisaccharide. So it includes the sucrose, maltose and raffinose. Polysaccharide is structural polysaccharides and the storage polysaccharides. So structure includes the cellulose and starch and insulin. Now this is the complete classification of carbohydrate. Now what is monosaccharides? The very first one, the very first classification, as you can see here, this is monosaccharide. isn't it? So this monosaccharide are what? It is a colorless, sweet tasting solids that shows oxidation. Clear? It shows the oxidations, esterification and fermentations. Clear? That means of if any sweet tasting solid that show the oxidations, that means of it's kind of simple sugar. When you keep any simple sugar in the atmosphere, what will happen? It immediately start melting. Not immediately, but it will take time, but it start melting, isn't it? Right? So it shows the oxidations, esterifications and fermentations. Due to why due to the asymmetric carbons, this exists in the different isometric forms. And that isometric form, that is dextrorotatory and labiorotatory. Clear? Dextrorotatory and there is a labiorotatory. Now, why there is a dextro or labiorotatory? Because it's simple, because they are this is D, right? So this is D glucose means it's a dextrorotatory, L glucose means a labiorotatory. Clear? And you can see the structure how, so these are the similar, but these are slightly changed, right? So this is the mirror image, isn't it? Like that. So when you keep a mirror in front of here, how it like exactly this. So this is a labiorotatory, this is a dextrorotatory. And that's it is called the isometric form. Clear? Okay. Now come to the fructose. What is fructose? Fructose is called the fruit sugar. So it is generally found in any fruits. So sweetest among the natural sugar. That means of natural sugar, it is more sweeteners. Glucose is called the sugar of body. That is blood sugar. Clear? And this is structure, glucose and fructose. So fructose is a fruit sugar and glucose is the sugar of body. That is called the sugar, blood sugar. Now come to the oligosaccharides. This is formed due to the condensation of 2 and 10 monosaccharide. That means that this is also monosaccharide, this is also monosaccharide. But when two monosaccharides are joined together with the help of glycosidic linkage, this is a glycosidic linkage, they form a bond, and that bond is called the glycosidic bond, and the water molecule is eliminated. So they form a oligosaccharides. Clear? So when two monosaccharides join together, it form a oligosaccharides. Now the example of this oligosaccharide disaccharide is, you can see here, maltose, sucrose and lactose. Maltose, how maltose is made up of? Glucose and glucose when combined, when two glucose molecule combine, it form a maltose. Lactose, glucose and glucose form a lactose. No, lactose is sugar molecule. So okay, what is maltose? It is also called the malt sugar, stored in the germinating seeds of barley. So when the seeds are germinated, barley seeds, oat, in that it is stored. That is called the malt sugar. It is formed by the enzymatic action on the starch, and it is reducing sugar. It is called reducing sugar. Similarly, there is a sucrose. Sucrose, it is also called the cane sugar or the table sugar. That what we had in our kitchen, that is a table sugar. It is obtained from the cane sugar, and beetroot on hydrolysis splits into a glucose and fructose. Clear? That is called the sucrose. And lactose, so all the milk sugar present in the mammalian milk on hydrolysis, if they yield glucose and galactose, Streptococcus lactic converts lactose into the lactic acid and cause the souring of the milk. Clear? So these are the maltose, sucrose and lactose, which is the product of disaccharide. So disaccharide is what? When the two molecules are combined with the help of glycosidic bond. This bond is called the glycosidic bond, right? So it is called the maltose, sucrose and lactose. Now, trisaccharides. Another example of trisaccharide, which is called the raffinose. So where this raffinose found? Raffinose found in the sugar beet, cotton and in some fungi. It is made up of glucose, fructose and galactose. So you can see here, this is glucose, fructose and galactose, and when all these three molecules are joined together, they form a raffinose. Okay, so keep in mind that glucose plus fructose plus galactose, it form raffinose. Clear? Now polysaccharides. Polysaccharides, now you know very well monosaccharide, disaccharide means when the two glucose molecule joined, so it form a disaccharide. Now polysaccharide means more than two. So general formula is C6H10O5N. N for is number of component joint formed by the condensation of several molecules. Either it can be 300 to 1000 of the monosaccharide is called the polysaccharides. Now on the basis of the structure, you can see polysaccharides are of two types: homopolysaccharides and heteropolysaccharides. Homo means same, right? So unbranched and branch. Unbranched now here means there is no any branching form in this. So this is unbranched homopolysaccharides. But this is what? This is the branch, and branch is forming over here, right? So this is a branched homopolysaccharides. Heteropolysaccharide, hetero means different. So two monomer types unbranched, like here see yellow and blue. So these are the two different monomers which are joined alternative. So this is called the unbranched heteropolysaccharides. And multiple monomer type or branch. So here you can see multiple means, now here you can see multiple means three, isn't it? These are called the multiple monomer type or branched. So homopolysaccharide, these are made up of polymerization of single kind of monosaccharide like starch, glucose, cellulase or the glycogen. Heteropolysaccharide means these are made up of by condensation of two or more kind of monosaccharides, which includes the chitins and the pectins. So chitin and pectins are heteropolysaccharides, and starch, cellulose and glycogen are the homopolysaccharides. Clear? Now on the basis of the functions, functions, there are certain carbohydrates which are stored in the different form, like starch generally stored in potato, right? So that storage is called the food storage. These serve as a reserve food, starch and glycogen. Structural polysaccharides, these take a part in the structural framework of the cell wall, like chitin, cellulase, as I told you, right? Plant cell wall is made up of from cellulase, fungi cell wall is made up from chitin, isn't it? So these are the basis of the function of carbohydrate. Now glycogen. Glycogen generally it is glycogen present in the animal cell always. Okay, so glycogen store the product in the blue-green algae, slim molds, fungi and certain bacteria. Starch, it is insoluble in water and give blue color when treated with iodine. Similarly, there is a iodine. It is a fibrous, rigid and insoluble in water, and it doesn't give any color when treated with the iodine. Right? So these are the three component: cellulose, starch and glycogen. Now, what are the functions? Cellulose, pectin and chitins are constituent in the cell wall of the higher plant. So what is the function? Simply, it form a protection. Clear? Peptoglycan in the cell wall of the prokaryote. That means of peptoglycan is present in the plant cell wall. In prokaryotic cell, not plant cell, but in prokaryotic cell it is present peptidoglycan. And in eukaryotic cell it is a cellulase, pectins and chitinins are constituent in the cell wall of higher plants. These are the reserve food material and form of protective covering. Fibers obtained are found in the making cloth and ropes. There are various type of fibers which is a part of only a plant cell, which is used for making the cloth and ropes. So like cloth, like all cotton plant, cotton cloth is an example of fibers, right? And there are certain ropes. Nitrocellulase and trinitrate cellulase, gun cotton, are used as an explosive. Explosive means generally used in making crackers and all, right? So it is nitrocellulase and the nitrite cellulose. Now, conjugated complex polysaccharides. So when I say conjugated complex polysaccharide, cell membrane is the example of conjugated polysaccharides, right? So here you can see the glycoproteins. Where is the glycoproteins? Here you can see the glycoproteins, right? So this glycoprotein is a conjugated complex polysaccharides. Example: blood antigen, collagen, lens proteins, blood proteins, hormones like FSH, LH, TSH and CGS. FSH means follicle stimulating hormones, LH for luteinizing hormones, TSH means, what is TSH? Thyroid stimulating hormones, and HCG is a kind of hormones which is used for I mean to say the menstrual cycle, right? And the cell membrane. So these are the different kinds of glycoproteins present in with their examples. There is a glycolipids like blood antigens, nerve fibers and cell membrane. Then there is a mucopolysaccharides, which is includes the heparins, then hyaluronic acid and synovial fluids. These are the mucopolysaccharides. So these are the example which you need to remember of conjugated complex polysaccharides. Okay, so we completed the complete carbohydrate. Simple, from started from the what is carbohydrate, what are the classification of carbohydrate, what are the function of carbohydrate, that we completed. Clear? And in short, you have to remember a simple, very simple example. Like here I'm just drawing this one. Carbohydrate classified into two types: sugar and non-sugar. Sugar is sweet in taste, non-sugar means it's a tasteless, amorphous solid, non-crystalline and insoluble in water, and sugar is a crystalline, soluble in water, right? This includes many sweets, monosaccharides on the acidic hydrophilus, but but sugar, which is sweet in taste, that is classified into monosaccharide, oligosaccharide, polysaccharides. So monosaccharide is aldose, ketose, and oligosaccharide is a disaccharide, trisaccharide and tetrasaccharide, which include the example sucrose, maltose and lactose, raffinose and dextrose. Clear? Is this clear? Okay. Okay. So now I'm going to give you break. for 5 minutes, and then we will start our session at 8:30. Okay, from lipids. So in the lipids, we will also learn about what is lipid, what are the structure of lipids, and we'll focus on the function also. So yes, you can take a break, and then after the break, we will start from the lipids. Clear? Okay. Hello. Yes, Arti.
Yes,
Shall
sir.
I start?
Yes, sir.
Okay. So now previously we completed the carbohydrate. Now we'll talk about the lipid. It in common way it is called the fats also, right? So simply lipids, fat is very common, like because it's not soluble in water, right? So any oil. So what are the typical example of lipids? All the oils, then butter, ghee, and all these are the kind of lipids, right? So fats and its derivatives are together known as a lipids. Fats and its derivative, that is called the lipids. Now this term lipid was given by German botanist Wilhelm Blur for fats and fats like substance. Means it fats and similar fat like substances are called the lipids. Now this lipids is constituent basically made up of from carbon, hydrogen, oxygen, but the ratio of carbon, hydrogen and oxygen is not 2:1. Clear? So these are the essential component which is involved for making the lipids. The amount of oxygen is considerably very less as compared to the carbohydrate. Now, as I told you that lipid is insoluble in water and soluble in organic solvent. So basically organic solvent means lipid can easily dissolve in acetone, dissolve in chloroform, benzene, hot alcohol, and even the ether. But it do not soluble in water, and that's why it is insoluble in water. Lipid do not form a polymers. Means never lipid form a polymers. Polymers means what? Like one chain joined with another one and they form a long polymers like this. So lipid never form a polymers. This keep in mind. Do not form a polymers. Lipid provide more than double energies compared to the carbohydrate. That means of like glucose. If glucose break down, it release just a single energy like 1 ATP. ATP, just one example, 1 ATP. But when the lipids break down, it release direct double ATP or double energy, and that's why lipid having constitute the more energies compared to the carbohydrate. Clear? So it is always double energies compared to the carbohydrate. Now this entire lipids is classified into three types: simple lipids, compound lipids, and the derivatives. Simple, compound, and their derivatives. Clear? Okay. Now, as I told you that the lipids basically insoluble in water but soluble in non-polar solvent, and that non-polar solvent is called the benzene, ether, and chloroform. Why it is soluble in polar form? Because they contain the long hydrocarbon chain that are non-polar, and thus they are the hydrophobic in nature. It is not hydrophobic, but it is a hydrophobic in nature, right? Sorry. Now come to the simple lipids. So simple lipid is what? Esters of long chain fatty acid with various alcohol, that is called simple lipids. Three molecules of fatty acid linked with one molecule of glycerol by ester bond, and such type of lipids are called glycerides. So this is a ester bond when this is ester bond when joined with the glycerol, they form a one glyceride. So this is our glyceride. Clear? This is a combination. This is structure of simple lipids. Now simple lipids contain two type of fatty acid: saturated fatty acid and unsaturated fatty acid. Right? Now you have to remember what is saturated fatty acid. The typical example saturated fatty acid example is lauric acid. Lauric acid, the very first one. Then there is a myristic acid. Myristic acid, the second one. There is a palmitic acid. Palmitic acid, and there is a stearic acid. Okay, so these are saturated fatty acid, which is very important that you have to remember also. Saturated acid with examples. Unsaturated acid, fatty acid, that is called the oleic acid. Oleic acid. No need to remember the structure, but you have to remember the name. Okay, so it's a oleic acid. Then linoleic acid. Linoleic acid. Then there is a alpha linoleic acid. Then there is a gamma linoleic acid. Gamma. Then there is a racinoleic acid. Racinoleic acid. Okay, so these are total 5 in number. So you have to remember what is saturated and what is unsaturated fatty acid with examples. So this is the example: lauric acid, myristic acid, palmitic acid, and stearic acid is example of saturated fatty acid. And unsaturated fatty acid includes the oleic acid, linoleic acid, alpha linoleic acid, gamma linoleic acid, and ricinoleic acid. Clear? These are the examples. Now, conjugated phospholipids. Conjugated phospholipids, it is made up of a two molecule of fatty acid plus glycerol plus hydrogen phosphate plus nitrogenous compound. This is called phospholipids. See the structure. What is the structures? The head, then this is the tail. Like this. So this region is called hydrophilic. This is hydrophilic in nature. Like this is the one which is a hydro, this one is a phospholipids. So this head is called the hydrophilic head because it always water loving. This region is a hydrophobic in nature, so it is a polar in, because it's made up from lipids, so it is a polar in solvent. So that means these are the hydrophobic in nature. So this one is a hydrophobic nature, and these are the hydrophilic because it is facing towards the external. This is also facing towards the internal. So internal and out and external cellular space are water loving, and in this middle, because it is a sandwich, so in this middle there is a hydrophobic. So these all are the hydrophobic regions. So phospholipids are the most abundant type of lipids in the protoplasm. So in all type of cell it is found. Phospholipids have both hydrophilic polar end and nitrogenous compound and hydrophobic non-polar end. So this is the non-polar end, right? This is the polar end. So hydrophobic non-polar end. So this is a hydrophobic, which is a non-polar. This is a non-polar, and this is the hydrophobic, which is a polar. Clear? Now glycolipids. Here you can see this is the glycolipids, structure of glycolipids. So it is made up of two molecule of fatty acid. Two molecule of fatty acid, then sphygnosine, galactose. It form a glycolipids. Example is which occurs in the white matter of the brain is an example of glycolipids. Means in our brain there is a cerebroside where there is a white matters are present. That white matters are made up from glycolipids. There is a sphingolipids. So in place of glycerol, they contain an amino alcohol sphygnosine, and these occurs in myelin sheaths of the nerve. So you might have seen the structure of neurons. Like this are kind of hairy structures, and then there is a nerves, and it is coated with myelin sheath like this, isn't it? So where you will get these sphingolipids? This occurs in the myelin sheath of nerve. So this is the myelin sheath, this is non-myelinated, and this is myelinated. So this myelinated sheath is covered with the sphingolipids. Clear? And that's why it is called the myelin sheaths. Now derivatives of lipids. So monoglycerides, diglycerides, steroids, terpenes, then carotenoids, they are formed by the hydrolysis of fats, and these all are the derivative of lipids. Clear? So that you have to remember that different derivatives of lipids. Now what are the important function of lipids? So basically oxidation of lipids yield comparatively more energy in the cell than the proteins and carbohydrate, right? So The oil seeds such as groundnut, mustard, coconut store fat to provide nourishment to the embryo during the germinations. So, like you know that the role of this lipids, right? Any seeds like any groundnut seed or any mustard seed or any coconut seed, they basically store the fats. And these fats, when break down during the germination time, it provide energy for the proper germination. Clear? Second, third, these functions are structural constraint, that is all the membrane system of the cell are made up of lipoproteins, and that's a structural component. It works as a heat insulator. What? Lipid work as a heat insulator and is used in the synthesis of hormones. So various hormone synthesis also take place with the help of lipids. Fats provide solubility to vitamin A, D, E and K. What is A, D and K? A is a vitamin A, vitamin D, vitamin E and vitamin K. All are the solubles, all are soluble in organic solvent, insoluble in water, right? So these vitamin A, D, E and K is going to solubilize in fats. So that means of it provides solubility to various vitamins, and all these vitamins are very very important for body. When you take that vitamins, it is not going to digest or not going to solubilize in your body until where there is a presence of fats. That means of fats provide the solubility to these particular vitamins. Clear? Okay, so after carbohydrate, lipids, the next is proteins. This is a third important chemical constituent, that is called proteins. Now, what do you know about the proteins? Yes, any idea? What do you know about the proteins? Simple. Yes. Proteins. These are the most abundant biomolecule of the living system, and it is the chief source of protein like cheese, milk, pearls, peanut. All are considered as a proteins. They form the abundant basics of structure and function of the life, and they are present in every part of the body, isn't it? And that proteins are required for maintenance and the growth of body, and that's why we need ample amount of proteins in our body, right? So here you can see proteins are the polypeptides. Polypeptides means it is a long chain of amino acid. Proteins is a heteropolymer and not a homopolymer. Homo means just a single one. No, it is see the combinations. So use combination. So it is a heteropolymers, and all these are not, though the color are same, but all are not similar. Okay, they are all are different. All these are the different proteins. Like some are simple amino acids. These are the different type of amino acid. It is not, all the amino acids are not similar, right? So the proteins is a heteropolymers and not a homopolymers, and the structure unit of protein is called the amino acid. So this one circle is called the one amino acid. So one amino acid, so there are n number of amino acids, and each amino acid is a amphoteric compound because it contains weak acid and a weak alkaline group, and that's why they are called the amphoteric in nature. Means there is acidic also, there is a alkaline also. Weak acidic and weak alkaline is there, that's why it is called amphoteric in nature. Now most of the amino acids are amphoteric in nature with one amino group and one carboxyl group. The insoluble nature of amino and carboxyl group into ammonia and carboxyl group make amino acid deuterionic form. This is important. Deuterionic form. So this is amino acid and this is their deuterion form. Clear? Okay, why you are moving? Because the camera is, slightly, yes. Yes, tell me.
So because I'm holding my tab in my hand, so it's a little bit shaking.
Okay, 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 all right. So, proteins. What is the exact number of proteins? There are total 20 amino acid in numbers. And these are specified in the genetic code. What is genetic code? It is a kind of amino acid dictionary. You know, the dictionary having lots of words. Similarly, in genetic code, there is amino acid, different amino acid have mentioned with the different codes. And these are universal in viruses, prokaryotes and eukaryote. Means in it is not similar to prokaryote, eukaryote and viruses. All proteins having a different, and that's why these are the universal which take part in the protein synthesis. So except glycine, amino acid has two isomer. That means of only glycine has only one isomer, and rest of all 19 amino acid are isomers. Eukaryotic proteins label L amino acid while D amino acid occurs in the bacteria and antibodies. Now, amino acid join with the peptide bond and form a long chain called polypeptide. So here you can see this is one amino acid, this is another amino acid, and it is going to join with a polypeptide bond and it form a polypeptide chain. Right? So this is dipeptide, dipeptide. Here it is a peptide bond and this is the water molecule. So these are the structures of proteins. Now amino acid is classified as I told you that they are amphoteric in nature, right? Amphoteric means what? That is weak acid and weak alkaline, right? So amphoteric, so that means of some are hydrophilic, some are hydrophobic in nature. So those amino acid which is hydrophilic in nature, they are positively charged, negatively charged or uncharged. What is the name of that amino acid? So those which are hydrophobic but negatively charged is aspartic acid and the glutamic acid. Aspartic acid and glutamic acid is a negatively charged hydrophilic polar water lover, we can say. Positively charged like lysine, arginine and histidine, these are the positively charged amino acid, and uncharged is a serine, threonine, asparagine, then glutamine, then cysteine and tyrosine. These are the uncharged hydrophilic polar amino acid. Hydrophobic non-polar. So aliphatic side chain and the aromatic side chain. Aliphatic, which include the glycine, alanine, valine, leucine, isoleucine, proline and methionine. And aromatic, which having some kind of fragment, I mean to say fragrance. So they are the aromatic side chain, which includes the phenylalanine and the tryptophan. Phenylalanine and tryptophan. Now here you can see this amino acid can be further classified on the basis of the requirement. Like suppose I want amino group. So amino group essential and non-essential. So these are the amino and which are essential. So when I say essential, lysine and threonine is essential amino acid and non-essential amino acid is a serine, glycine and cysteine. Non-essential amino acid branch chain with amino acid, tryptophan, then phenylalanine and methionine. These are the essential and these are the non-essential, which includes the glutamate, then alanine, aspartate, then glutamine, asparagine, proline, tyrosine. These are the non-essential amino acid. Similarly, there is a histidine is also common as non-essential histidine, then arginine, serine, and taurine. Now, come to the structure of proteins. How exactly, what are the typical structure of proteins? As you know that it is a chain of long amino acid, right? So here it is called, suppose this is a single chain, this is called primary structures. If the structure is somewhat like this one, zigzag, or somewhat like this. One is going up, another going down. So leak-lock pattern, or we can say alpha helix. These are the secondary structure. These are the secondary structure of proteins. Tertiary structure means more condensed, as you can see. And quaternary structure is, so this is also referred to the 3D structures. And these are more condensed, which is called the quaternary stage. So proteins are classified into 4 configuration. They are of based on their configuration. It is of 4 types: primary structure, secondary structure, tertiary, and the quaternary structures. Okay. Now what is happening in the primary structures? So basically in primary structures only peptide bonds are present between the amino acid. So that means of this is one amino acid, this is another amino acid. So in between these two amino acids there is a peptide bond. Similarly, this is amino acid, another peptide bond. This is one amino acid, this is another peptide bond, like this. So bonds are present between the two amino acid. The first amino acid is called the N terminal. Like here, this is the first one amino acid, so I can say this is the N terminal. This is the last one, so I can say this is the C terminal. Clear? So first is N terminal, C is the last C terminals. Amino acids, such proteins are not functional. So there are certain which proteins are not functional, so they are come under the C terminals. Clear? So that is simple primary structures. When it says secondary structures, so the protein molecule of secondary structures are spirally coiled, means the same things, but it is highly spirally coiled. Can you see here? Yes. So this highly spiral coiled and stabilized by the straight hydrogen bond between the amide groups. So this secondary protein structure is classified into 2 types: beta pleated sheet and the alpha helix. So beta pleated sheet like this means, suppose one is going up, like here you can see this diagram is a beta plated sheet and this diagram is called the alpha helix sheet. Clear? To 2 types. Now tertiary structures. So proteins of amino tertiary structures are highly folded, means it is more coiled, highly folded. As I told, this is a 3D shape, look like it's a 3D shapes. So that's why it's called tertiary, second degree structures, third degree structures, right? So proteins of tertiary structures are highly folded to give a globular appearance. So the structure is called the globular appearance, and these are soluble in water. Clear? So here you can see this is the ionic bond, and this is called the disulfide bridge. That means of this bond is basically joined. That's why it is a disulfide. Sulfur group is over here. So this is a disulfide bond. This is the ionic group. So the ionic bond is over here. This is a hydrophilic interactions, and this is a hydrogen bond combinations, like here. So that's why it is soluble in water. The last one is a quaternary structure. So 2 or more polypeptides chain of tertiary structure united by different type of bond to form a quaternary structure of the proteins. So here you can see that this is the hemoglobin is one kind of quaternary structure of proteins, which includes the oxygen molecules, the polypeptide chain, then iron, then heme group. Everything is there. That is called the hemoglobin, which is a quaternary structure of proteins, and quaternary structure is look like this only. Okay. Now, as I told that proteins are further classified into simple, conjugated, and derivatives. So globular proteins includes the albumin, globulins, then gluteins, prolamines, then histamines, globins, and protamines. Then scleroproteins include collagen, elastin, and keratins. Conjugated nucleoproteins, then glycoproteins, mucoproteins, lipoproteins, then there is a phosphoproteins, chromoproteins, metalloproteins, right? But these simple and conjugated is quite, we can say that different in structures and nature, but derivative proteins are primary and secondary, which include the coagulated proteins, then proteons and metaproteins. then proteases, proteins, and polypeptides and peptides. So these are the different based on their chemical compositions, they are classified. Now, what is the function of proteins? So formation of cell and tissue for the growth, that is the first function of proteins. Formation of hormones and enzymes. Then for muscle contractions like actin and myosin, help in the blood clotting. For transport, hemoglobin and transferrin. So basically all these functions performed by the proteins. For defense against the infections like antibodies production and all. Then for storage, myoglobin and ferritins. For supports like collagens and elastin, these are the different functions performed by the proteins. Now, proteins. Inside there is a nucleic acid. Now, where you will get the nucleic acid? As I told you that DNA is the building block of all the organisms, right? So here you can see this is the DNA molecule and this is the RNA molecules. So where this nucleic acid present? So nucleic acid is present in all the cell and virus. Okay, they are polymers of nucleotides and hence they are called the macromolecules. So this nucleic acid, Michecher discovered the nucleic acid in the nucleus of pus cell and later he called it as a nuclein, and the name nucleic acid was proposed by the Altman. Okay, so Michecher and the Altman is there. Another one, on the basis of the structure, the nitrogen bases are broadly classified into two types. One is called the purines and the pyrimidines. Now pyrimidines is cytosine, thymine and uracil. Cytosine, thymine and uracil is pyrimidines and purines is adenine and guanine. So here you can see the structure. This is adenine, guanine. You can also remember that nitrogen bases is arranged at which locations. So how I can count this to understand this? So first of all you have to count from here. This is 1, 2, 3, 4, 5, 6, 7, 8, like that. Okay, so this is 1, 2, 3, 4, 5. Sorry, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, like that. Okay, so that you have to count it. I mean to say you have to remember that nitrogen bases are present at which location. That is important. So this is a similar structure that adenine and guanine. Similarly in pyrimidine, so there is a one benzene ring and there is a amino group, not nitrogen. It is amino group attached to which position that should be there. So in a cytosine, uracil and thymine, these are the structures. So structure, remember, is very important because sometimes this structure play a very crucial role to identify a particular group of amino acid. Yes, nucleic acid. Is this clear? Okay. Okay, just a minute. All right. Now, nucleic acid. You know the structure of DNA, right? In 10th standard also you studied about the structure of DNA. Now here you can see a nucleic acid contain deoxyribose, and that's why the name is deoxyribonucleic acid, DNA. So who first time discovered this? Wilkins and Franklin studied DNA molecule with the help of X-ray crystallography, but the Watson-Crick proposed the double helix model of the DNA. So this is the Watson-Crick model, double helix of DNA, which includes the major groove, minor grooves, and their size from here to here is 3.4 angstrom unit, and this is 0.34 angstrom unit. In between the two amino acid, it is 0.34 angstrom unit. Clear? And this distance is total from here to here. It is 2 nano angstrom unit. Clear? So these are the structure of nucleic acid. Now this nucleic acid, as I told you, these are the double helical structures. One is going up, one is coming down. So it is anti-parallel and double helical structure. This is the major groove. This region is called major groove. This region is called the minor groove. Means little depth is there. This is more depth. So this is the major grooves. Nitrogen bases are present on each side. These are the nitrogen bases present over here. And that nitrogen base is in the form of adenine, guanine, thymine, cytosines. And when you see this same structure in what we can say in a very complex form, then these are the phosphate sugar. Here you can see this is the pentose sugar, and here you can see phosphate group is attached. PP stands for phosphate group attached. This is phosphate group, this is phosphate group, and these are the nitrogen bases. So you can see thymine, then there is glycine, then cytosine. These are the nitrogen bases. Adenine, then cytosine, and guanine. This is another nitrogen base for this helix, and this is for this helix. Clear? So these are the anti-parallel strand with the nitrogen bases and represent in a short form. So these are the shortest form of representation of the amino acid structure of DNA. Now nucleic acid, this DNA is of various types, like there is a bDNA, zDNA, aDNA, cDNA, and ddNA. Okay, these are the types of DNA. When I say what is the type of DNA, what is a bDNA? So when I say bDNA, it is handeds of helix. So helix is always right-handed. Pitch of the helix per turn is 34 angstrom unit. Diameter of the helix is 20 angstrom unit, and stability is stable and physiologically active form. zDNA is a left-handed helix, 46 angstrom unit, and diameter is 18, which is more thinnest, and it is unstable. Clear? So in this, important is this one, AZ and cDNA. These are the important. Okay. So similar, so you can remember, see, right-handed, left-handed, all are right-handed. So it is very easy for you to remember that helix except Z, B, A, C, D, all are the right-handed, isn't it? So in this way you can remember the different types of DNA. These are their base pair, like base pair per helix turn is 10, here 12, 11, 9.338. So you have to remember the maximum and minimum. So maximum is 12 and minimum is 10. Clear? Now repeating unit is mononucleotide, this is dinucleotide, rest all are mononucleotide, only zDNA is a dinucleotide, rest all are the mononucleotide. Isn't it? Now what is the function of the nucleic acid? So the function of nucleic acid, DNA is vital for all the living beings. So it is important for inheritance. You know that DNA is the genetic material, isn't it? So that's why it is inherited from one parent to the next, one generation to the next generations. So it is coding for the proteins and the genetic instructions guide for life and its process. DNA holds the instructions for an organism's or each cell's development and reproductions, and ultimately the death. That means of DNA play a very crucial role in the any living organisms. Now nucleic acid, RNA, as similar to DNA, there is a RNA, but RNA is found only in viruses, right? So RNA, and in the eukaryotic cell, it is when there is a transcription, there is a mRNA, right? So RNA is a structure Structure of RNA is fundamentally the same as a DNA, but there is the same differences. Some differences there. These differences are in the place of deoxyribose sugar of DNA, ribose sugar is present in RNA. Like there is a deoxyribose because there is a deoxy present, and that's why deoxyribose DNA called. But here only ribose sugar is present, that's why it is called RNA. The first difference. In the place of nitrogen bases, thymine represent the DNA and nitrogen use uracil in the present in the RNA. That means of in the DNA there is adenine, guanine, thymine, cytosine, right? Isn't it? So in case of RNA, adenine is there, thymine is there. In the place of adenine, guanine is there, but in the place of thymine it is replaced and called it as a uracil. And cytosine is similar. So there is a AGUC. This is RNA and nitrogen bases, and this is the DNA nitrogen bases. Is this clear? Because it's saying here, see, in the place of nitrogen bases, thymine present in DNA and the nitrogen basis uracil is present in RNA. So this is the thymine present here. This is the uracil present over here. RNA is made up of only one polynucleotide chain, but RNA is single stranded. So here you can see this is a single stranded made up of from one polynucleotide chain. So RNA is single stranded. Now types of RNA. So here you can see there is a messenger. There are total 3 types of RNA. There is a messenger RNA which is called mRNA. There is a ribosomal RNA which is called rRNA, and there is a transfer RNA which is called tRNA. Okay, messenger, ribosomal, and RNA. So how you will remember in a very short form? Simple. You can just write it down. mRNA, tRNA, and rRNA. So M for messenger, T for transfer, R for ribosomal. So MTR you have to remember, remaining is RNA. So total 3 type: mRNA, messenger RNA, transfer RNA, and ribosomal RNA. Okay. So we completed carbohydrate, proteins, lipids, right? And nucleic acid. RNA also included, DNA and RNA. Yes. You can unmute. Aarti, you want to say something? If you want to say something, please. Okay, just tell me any doubt.
No, sir.
Okay, so shall we move on question practice?
Yes, sir.
Okay. So here the very first question, a typical fat molecule is made up of. A typical simple fat molecules is made up of 3 glycerol molecule and 1 fatty acid molecule, 1 glycerol and 3 fatty acid molecule, 1 glycerol and 1 fatty acid molecule, or 3 glycerol and 3 fatty acid molecule. No idea?
No, sir.
Okay. See what I said, simple fatty acid, right? Okay. So it's a very simple question actually. Oh my God. Again, who is asking? Okay. See the diagram we have already studied here. Can you see here? This is triglyceride. So how triglyceride is made up of? It is made up of from 1 glycerol and 3 fatty acid, right? So the fat is glyceride, which is made up of 3 molecule of fatty acid and 1 molecule of glycerol. So here you can see 3 molecule of fatty acids 3 molecule of fatty acid and 1 glycerol. So B is the right answer. Here 1 glycerol, 1 fatty acid, no, can't go. 3 glycerol, no, and 3 fatty acid, impossible. 3 glycerol and 1 fatty acid, no. So this is more accurate one, isn't it? Select the incorrect statement regarding the proteins. You know that we studied that different statement regarding the proteins. So 20 types of amino acid are used in the formation of proteins. Yes, can we say? We studied about the same. Another one, okay, no, we have to tell which one is incorrect, right? We have to tell which one is the incorrect. So this is the right one. Now go for another questions. Each amino acid is amphoteric compound because it contains one weak acidic groups and the weak carboxylic group. Yes, amphoteric is because of the same. So this is also the right statement, isn't it? Okay, go for the another one. Collagen is the most abundant proteins of animal world. That is also right. In protein, amino acids are joined by N-glycosidic bond. Is it the right one? No. In proteins, amino acids are joined by the hydrogen bond, right? Not a glycosidic bond. This glycosidic bond is present in the fats, lipids, isn't it? And that's why this statement is wrong, which is the right answer. Because he's asking is incorrect statement.
Option D, sir.
Isn't it, Aarti?
Option D,
D is what?
incorrect, sir.
Incorrect. Yes, absolutely. Okay, so what we studied the biomolecule, which include the lipids, nucleic acid, carbohydrate, and proteins. These are the different structure of proteins, carbohydrate, lipids that we completed. So total, how many biomolecules we completed? Lipid, nucleic acid, carbohydrate, and proteins.